Hello, good morning, everyone. I'm Julio Belmonte, here to talk to you the whole day today about first cell compartments and then about focal point plasticity or links, spring links between cells. So this is a picture of things that you'll be actually be doing today. And let's start with model 6.1, which is about cell compartments. Okay. So what you can see and maybe already kind of guess when you talk about cell compartments, you talk about modeling cells in CompuCell made of different parts, okay? And you can see two examples here on the left, on the right. A cell made of membrane, nucleus, and cytoplasm, and a cell made of three different parts. We're going to talk about how it works, why we want to do that, and the motivation. So we have, and what you see on the background is going to be the first model be exploring a model of cell migration. Okay. So again, if you have any questions, please use the chat. I'll try to read your question back and answer to you as best as I can. All right. So these are going to be the topics that we cover today. We're going to go a little bit of motivation, talk about structural and functional cell heterogeneities, internal and external contact energies. This part here I forgot to upgrade. Actually, we're talking about a cell with a nucleus, a membrane, and a lamellipodium. Okay, nucleus, cytoplasm, and lamellipodium. Not. I'm going to change the model, and then we're going to talk about creating a cell epithelium in CompuCell3D. Okay. And for all of them, we're going to talk about initial layout, how to set cell compartments, and how to choose the values. Okay. Okay, so this is going to be the new topic. All right, so why we want to model cells with different compartments, or why we want to model cells uh, made of more than one part? And the answer is simply because cells are not just a blob of amorphous material, right? The cell has heterogeneity within it. Okay, it has for example, it has a nucleus, has a lot of different compartments inside. It has a Golgi apparatus, has a mitochondria, has a membrane, a cytoplasm. They have very different properties. The nucleus may be very important, especially if the cells start to crawl in a crowded environment. The nucleus is harder to compress, so that may be an issue, right? So it may slow your cell down. When the cell is moving, right, actually, it has clearly a front and a back a side to it. It has a lamellipodium on the front, or maybe a filipodium on the front, protruding and crawling on the substrate or across other cells. So you have to, the properties of your cell when you model CompuCell3D, you probably want to make it different in the front versus in the back, right? And again, when the cell is crawling, the nucleus might be an important factor. So it would be a good idea actually model the nucleus explicitly in your cell. And finally, when your cells, usually they are part of an epithelium, right? They have an apical part, a basal part, right? And they have a different addition properties between with the ECM uh, versus the lumen and between the cells. In principle, you can model that just by changing the cell addition properties, but that's not very stable. But the cells also have other different properties on the basal versus the apical side and the lateral sides of the core side. So you probably also want to model uh, each one of those compartments more explicitly in your model, right? Maybe you want to have maybe your model cell as a cytoplasm, a nucleus, and a thin membrane composed of three parts, a membrane that is the apical membrane, the basal membrane, the lateral membrane. That may make sense for some applications. So CompuCell allows you to actually break up your cell into multiple parts and control the parts of each one of those parts explicitly, which make your model more realistic and make your model work better for specific environments. Okay. So of course, when you are talking about breaking your cell into multiple compartments, right? There are two ways to think about that. We can talk about either about structural or functional cell spatial heterogeneities. Okay, so what I mean by structural. On the structure is the one on the top on panel A, you're actually modeling different structures inside the cell, right? So you can model the membrane explicitly, you can model the cytoplasm explicitly, you can model the nucleus explicitly. Maybe you have to model the Golgi apparatus explicitly or the mitochondria of your cells explicitly. So each region in the cell corresponds to a very well-defined cell structure inside the cell. So that's what I mean by modeling the cells with structural heterogeneity. Okay. But sometimes the cells have a spatial heterogeneity that's not necessarily linked to a specific cell organelle, right? It's not the membrane, it's not the nucleus. Maybe it's more like the cell is polarized and one region of the cell behaves different from the other region of the cell because of collection of proteins and maybe cytoskeletal structure or other signaling events that make one side of the cell different from the other side of the cell. But it's the boundary line between when this region ends and the other region starts is a little bit uh, more arbitrary on your part as a model to say where one starts, where one ends. So this is the case, for example, when you model cell, an epithelial cell layer, where the cells have an apical, a lateral, and a basal side. Okay, clear the apical side of the cell has different properties than the basal side of the cell, right? And exactly where the apical region ends and where the basal region ends, a little bit more arbitrary. Maybe you only have the cell of apical and basal region. Sometimes you have to add the lateral region. Sometimes you even have to add the lateral region and a nucleus. So maybe actually you can mix both ways of compartmentalizing your cells. But this is a case where it's more a functional region than a really a structure, not something that you can map into an organelle. So you can use the cell compartments idea in CompuCell to model those two types. Okay. So has this ever been done before in CompuCell? Yes, many times. Actually, the first time that this was done was in 2007 by the group of Andreas Deutsch, where they model myxobacteria, right? And this is a little bit of mixture of functional and structural modeling. They have this cell made of different compartments. They have a front end and a back end, and have all these different cell regions here that just made the body of a bacteria. And you link them with some kind of spring forces, and then there's one side, the head can move in a specific direction, and the other places just follow. So that is an example where you use the idea of cell compartments. Okay, this model was not done in CompuCell, but you have to use compartments to actually get the correct shape and behavior of the cell that you want to model, right? So you cannot make such elongated and curving bacteria just using one cell compartment. You have to use many of them to actually capture this cell shape and behavior. Another one that we did a long time ago, Dr. Abbas Shinfrad, when he was still a graduate student with James Glazier, he used this idea to model an epithelium that invaginates and forms a new vesicle, right? A cup, eye cup, I guess that was the idea behind this model. So each one of those cells actually made of three compartments, right? This blue, the green, the red compartment. There are some forces between the red compartments, and those compartments can also change their size. The blue becomes bigger, the red becomes smaller. And all this together leads to this kind of invagination event and budding out of this new epithelial ball that you see here in a two-dimensional cross-section. So that was actually the formation of the eye lens from an epithelium. So that's another example of how it has been used. Another one, this actually by me, we use this idea of cell compartments to model the kidney tube where the nephron kidney too, where a cell becomes sick here and buds out and make a cyst. And in these cells here in this tube, they actually explicitly model the apical compartment, the cytosol, the base compartment, and the lateral compartment you cannot see in this 3D video, right? But you can see here in this cross-sectional, this cell is made of 4 compartments, and that's another example where I could change that. And the idea behind that is that the base compartment can secrete ECM, the lumen compartment, the apical compartment stays with the lumen, it can secrete more lumen, and I can change the properties of the lateral compartments to change for example, the type of cadherin that the cells expresses. So this was another example of where we use cell compartments. Okay, and you can see that this works actually also in 3 dimensions. Okay, another example, you can use the idea of cell compartments, use these kind of regions to model, for example, planar cell polarity. In this case, you're still dealing with an epithelium, but you're not looking at the epithelium on a cross-sectional. You're looking at the plane of the epithelium, and your regions are the proximal distal regions of the cell at the apical site. And they are initially mixed, and in this model, they kind of sort out based on the adhesion energies that they have with the other cells, and they become polarized along one direction. There's a preference between this red and green compartments, so the cells they sort out, and they change the orientation of the proximal and distal part, the red and the green part, and they all become eventually aligned over time. Okay, so that's another example where you can use cell compartments to model a process. Okay. In this case, the compartments are actually moving dynamically with respect to their neighbors instead of being fixed in space. Okay. Okay. So these are our examples, right? And again, we will work a little bit on both structural and functional heterogeneity in this example, and but we start with the structural spatial heterogeneity because it's a little bit easier to understand. Okay, so when you use structural compartments in CompuCell3D, right? You can do that for a resting cell or maybe for a crawling cell, okay? So when you do that for a resting cell, maybe one thing that you can do, you might maybe want to model the membrane, the cytoplasm, the nucleus, for example. If you have a moving cell, a cell that's crawling around substrate, maybe your compartments, you have to make a different choice of compartments. Maybe you still want the cytoplasm, the nucleus to be there, but maybe instead of modeling the membrane all around the cell, it's more interesting for you to maybe model just the lamellipodium of the cell that actually crawls on the cell. So in this case, the lamellipod is a clearly visible structure. Well, exactly where the lamellipod starts and ends, a little bit of the debate, but you just, in your model, you kind of just make, okay, there's going to be a compartment, there's a lamellipodium, a compartment is going to be the rest of the cytoplasm of the cell. So this is a little bit of structural and heterogeneity, structural and functional compartmentalization of the cells, but is you can say that's maybe structure because the lamellipod is clearly different where you have all your actin fibers being branching and polymerizing. So it's a clear distinct structure on the cell, although the boundary between where it exactly starts is a little bit fuzzy, but it's a clear visible structure. Okay. So this is going to be just 2 examples of modeling cells with compartments, and you can make a different choice of exactly what the compartments mean and what compartments to put in there or not put there. So you can have more, you can have less, maybe you can only have a cell and a nucleus, or maybe you can even have a chrome cell, just have the cytoplasm, the lamellipodia, right, and get away from the nucleus. So it's your choice as a modeler to exactly how many compartments you're going to put there and what they will correspond to. Okay. So far I have just me talking. Do you guys have any questions or comments before moving into the first exercise? Okay, so let's start the first exercise. What we're going to do, we're actually going to use a model of the cell crawling as a first exercise. So let me explain first how this model works. Okay, so this you see on the right, a video of cell crawling model. This actually is a model developed by Gilberto Thomas, who have already seen before. Okay, and this model, the cells have three compartments, right? They have a nucleus, a cytoplasm, and a lamellipodium. And what's making this cell move is that there is a external potential plugin being applied on the lamellipodium of the cell, okay, at specific direction. And so that's what makes the cell move in a particular direction. But you see that the cell actually also change directions, right? This happens because every now and then, every n steps, in multiple steps, you randomly choose the direction of this external force plugin that's applied to the lamellipodium compartment cell to a new random direction, and that's what causes this cell to going up and then change direction, okay? To another side, maybe go to another side. So that's basically what this model is doing, okay? And for this model, we use compartments, okay? We basically uh, use three compartments, and there are special instructions of how the compartments behave. And one thing that you may be asking yourself is why you're talking about cell compartments, what you make why it's important to uh, talk about cell compartments in this case. The importance of the idea of cell compartment is not just, okay, let's use many different cell types. Is actually something that you have to change internally in your code, because you have to tell the cells which compartment, which cell they belong to, okay? So if you just use cell types to do this, it worked very well for a single cell. But if we are not careful, the problem is that if you only use cell compartments, that eventually those two cells that are crawling around there, they're going to meet. And once they meet, there's going to be some sort of confusion about who belongs to this, who this nucleus belongs to, who this lamellipodium belongs to, and who the cytoplasm belongs to. And the cells will fuse and bad things will happen. Okay. So to prevent that, you have to tell the cell, okay, this nucleus belongs to this cell, this cytoplasm belongs to this cell, and this lamellipodium belongs to this cell and not the other. Okay. Otherwise, you're going to mix. Okay. So that's why you have to specify two cell contact energies, one for the compartments belong to the same cell, and another set of contact energies to the cells belong to different cells. And then here you see another more drastic example of 4 cells. So the more cells you have crawling, the worse the things you get. The cell, if you don't specify which compartment belongs to whom, they're gonna all mix together. It's gonna be a confusion. Okay, and this is certainly not a good model of cell crawling, because we are not respecting the individuality of each cell. Looks like they're dancing. Yes. Okay. So there was a question here in the chat. So in this model, the cells change direction randomly after any steps. Yes. Can we model a cell that change direction if it encounters an obstacle? Yes, you can. That's going to be you certainly can. You need to have some kind of criteria to identify when the cell hit an obstacle. Maybe the cell stops moving in the direction that it wants to move, and that's going to be a sign that it encounter an obstacle. And then if it's not updating its direction, in the direction of the movement, maybe it's time for you to choose a new movement. So maybe you can, that may be the criteria. But yes, you can do that. But the focus here now is just for you to first learn how to make this model and make it behave such a way that this kind of fusing event or dancing of the cells very close to each other will not happen, okay? Or playing football, okay? Okay, uh, so the idea here is actually for this to happen, you have to specify cells have make a differentiate between external contact energy between the cells and internal contact energy between the cells, okay? So your cells will have two sets of contact energies. One, when the cells interact with other cells. So when you have a nucleus compartment interacting with another nucleus compartment or the cytoplasm compartment of a different cell, so this is going to be all the interaction of energy between those compartments with other cell compartments. And you also have a ContactInternal plugin, which is going to be all the energies of these three regions within the same cell or cell cluster, okay? How does CompuCell know when it's a compartment belong to one cell to the other? Let me just skip my slides here and tell that this is done using the idea of the cell IDs and cell cluster IDs, okay? So let me explain what the cell ID is. I do not know if you have seen, but every cell in a CompuCell simulation has an ID, a unique number. You can think of it as a social security number that's unique for each one of you, okay? So, for example, in this model here of somatogenesis, you have those cells as green, red, and blue cells. And it doesn't matter which type they have, each one of those cells have a unique cell ID or a unique social security number. This cell has ID 1, this red cell has an ID 2, 3, 4, 5, 6, and so on. And it never repeats, and every time you create a new cell, it receives a new ID, or every time someone is born in the United States, it receives a new social security number of someone comes to the United States and becomes citizens. He also receives a new social security number, right? So every cell in the CompuCell simulation has a unique ID, and when the cell dies, the cell ID is deleted and not used again, okay? When you're using your cell compartments, each compartment of the cell also has this unique ID, right? So for example, in this crawling cell, the lamellipodium has an ID 107, an ID, cytoplasm has an ID 32, and the nucleus has an ID 24, okay? But how do you know that all the cells belong together? That's because you have another ID number that specify which cells belong to each other, which compartments belong to the same cell object. Okay, so this other ID is called the cluster ID. Okay, so in our CompuCell simulations, each cell has two IDs, one that specifies who they are, and another one is called the cluster ID that specifies to which cluster of cells or which they belong to. So for example, in those two cells here, for example, this cell has 24, 22, 43 IDs, but all those compartments have a cluster ID number 2, so that they know that they belong to the same cluster, okay? And all the cells here, they have IDs 59, 88, 85, okay, which is unique. But in addition to that, they have a cluster ID 10. So these lamellipodium, cytoplasm, and this nucleus know that they belong to this cluster of cells and not to this cluster of cells. Okay. So there was a question, when the cell divides, two new IDs are created or one is kept for one of the other cells. I think when the cell divides, and please correct me, the people that know the code better, one of the cell keeps its current ID, and a other cell will receive a new ID. Okay, so that's what happened. You just create one new ID. You do not get rid of the last one. Okay, so when you do the cell division, I believe you have already done that. You may notice that you have a parent cell and a child cell, right? The parent cell keeps its ID, and the child cell receives a new ID, okay? So it's more like a birth process, right? When the woman gives birth, right, she keeps her social security number, right? She does not become a new person, but the baby is a new person, so he receives a new social security number. So you can make this analogy. Is a new daughter ID increasing the order creation? I guess it is. Okay. I guess the computer looks at the what next available ID and use the next one. Okay. Yeah, ideas are sequential. Okay. The ideas are sequential and IDs are unique. But you can notice that the cluster ID is assigned sequentially, right? If you don't say anything, but they are not unique. Multiple cells can have the same cluster ID. That's what makes the cells gives the information that the cells belong to the same cluster or the compartments belong to the same cluster. Okay. But the cluster ID can change over time. You can suddenly say that, okay, from now on, this lamellipodium will detach from the cell and receive a new cluster ID. Okay, so this is a number that actually can change over the simulation. Okay, so you can make two cells merge, you can make two cells separate, so you have more freedom to change the cluster ID of the cells. Okay, so that is how you keep track inside CompuCell which compartments belong to the same cluster and which compartments pairs belong to different clusters. You just check their cluster ID, and if they're different, you use the contact energies. If they're the same, then you're using the internal contact energies. So it's by this tracking that you know whenever you're looking up over this table here or over this table here on the right. Okay, so it's here on the slide. So this sets the contact energy between internal compartments or the cells, the compartments that share the same cluster ID, and the left one sets the energy between the cells that do not share the same cluster ID, so they belong to different cells. Okay. So how does it work? So this means that you have to set different energies between for the contact and the contact internal, right? So that means that you have to make a wise choice of which energy to choose. Otherwise, your simulation is going to look very bad, okay? So for all this, so what you're going to do here, you're going to grab the cell sorting. So the crawling model that you have seen for this video here, right? And this model is available on the file 6.1.1a. You can download it from the student materials. If you have not done so, please go to the student materials and and download 6.1.1 file, okay? And we're gonna use that, and you're gonna use that as a starting point to make that model to behave as a use that starting model to make it work well, to make as a compartment and make the cells grow and divide. Okay, so that is the idea of the exercise here. We're gonna download the file 6.1.1. So right here on the chat. So please download file zip and open in Tweedit. Okay. Okay, and I'm gonna implement the model that you have here. Right now, the model on 6.1, it only has one cell that moves. It has not multiple cells. We're gonna change that to make it have multiple cells that do not merge with one another. Okay, so in another word, you're gonna change the model to make it behave, to make the correct implementation of the cell compartment idea. Okay. Right, so please, and then that's gonna be our objective. So has everyone downloaded the model and opening Tweedit? If you have done so, please let me know on the chat, and then we can start with the exercise. Okay. So for now, we just download the 6.11a and unzip and open it. You don't need to do anything on the file, just look at it, and I can explain what's happening there. Sorry for the confusion with the zip folder. it creates two folders, right? Yes, as Noshina Fatima said, there's a file called e4.122.cc3d. That's the CompuCell file that you have to open. 20 people has already downloaded the file. Let me just open the file and show how it looks like. So here's the file that you should see, right? And just let me give a quick go through how it works. We have the XML file, right? The Potts file, and then here the cell type. You see that's made of 3 cell types: cyto, lamellipodium, nucleus. It has a volume. It has an external plugin that makes the lamellipodium moves. Okay. You have the contact energies of the 3 compartments, which are not. So notice that you only have a contact plugin doesn't have a contact internal plugin, so we're going to change that, right? And it has a pre-specified additional energies that it works well for this model, and you initialize the cell types one close to the other. You just one of each, and here on the Steppable file, you see that on the start function you only have one Steppable. In the start function, you just define this volume of each one of them, okay? And the lambda volume, okay? And on the step function, that is where you select the cell lamellipodium, right? And if the cell type lamellipodium, you apply the external plugin, and you change each value randomly, the x and y direction, and you do that at a given time interval. And this time interval is every 1,000 Monte Carlo steps. So basically, that's how it works. I just give a quick go through about from the model, okay? And basically, that's how the model works, okay? So you don't need to run the model right now. If you want to run the model like Ronak did, probably you get hopefully a working model. You see them all moving in change directions, okay? So you see that this is a perfect fine model when you just have one moving cell, okay? But the idea now is that now we won't have multiple moving cells as you see in this movie here, right? So if you want to do that, you have to tell the cells that they all belong to the same cluster, right? Before you can grow them, and before you can divide them, and before you can have multiple ones. Because if you don't do that, instead of have this beautiful movie that you see here, you have this messy movie that you see here, okay? So let's go through about exactly how we're gonna go about it. Okay. So let me go back to the slide. Okay, so we have the contact energies and the contact internal energies. So before you go into the code, I want to just talk you through in a theoretical level of what we should do, okay? So first thing, you have to now have the contact internal energies, right? Because we have to treat them differently. Now, for the contact internal, that's going to be the energy between the cell, the nucleus, the cytoplasm, and the lamellipodium, right? And in your contact internal table of energies, right? We can just use the original values of the original code, right? So they have it at 11, 10, and 40, okay? We can just use those, okay? So we can just use the same as before, okay? But you have to move to the contact internal plugin. okay? The other thing that we need to do, right? You notice that a cell can only have one nucleus, one cytoplasm, and one lamellipodium, okay? So that means that you can pretty much ignore all these diagonal terms here, okay? Because you only have one of each by definition each one of those cells. So you can skip the definition of this cytoplasm, lamellipodium, nucleus, contact energy internally because the cell have only one copy of each. In some models, you may have a cell that's multinucleated, but in this case, you need to specify the interaction of nucleus and nucleus that belong to the same cell. But it's not the case here. So basically, our ContactInternal table is basically done, okay? By definition, the medium is never part of a cluster of cells, so you can also pretty much ignore all the internal contact energies with respect to the medium because the medium is not part of the cell, is not part of the cluster, okay? You cannot assign a cluster ID to the medium, okay? So you can pretty much ignore that, which means that the interaction of all those compartments in the medium have to appear on the contact, the external contact energy, right? So that's where those goes. So here we can use the original values for the interaction of all those compartments in the medium because medium is always an external entity, right? By definition, it's always something external to the cell. So that's where all your external energies go. Is going to be going to the main contact energy table. So here you can just use the model that we had before, 22, 10, and 100. Okay, so the nucleus has a very high energy because you never want the nucleus to touch the medium, right? So that's why it's very high. The cytoplasm and the lamellipodium, they actually, it's okay for the contact the medium. So that's why their energy is lower than the one in the nucleus. So the compartments, they have to be completely inside the cell. They have to have a very high energy briefing that's external. And the compartment, they actually touch the external environment. They have a lower energy with this environment. But as a rule of thumb, when you use compartmental cells, is that all the external contact energies have to usually be higher than the internal contact energies, okay? Because that's what's going to make your cell cohesive, right? The energies between the internal compartments usually have to be lower than those same energies of the external compartments. So usually those values here on the table for the external contact energies are usually bigger than the energies in the internal contact energies, unless you want to have two compartments that never touch each other. For example, you want the nucleus to be on the back and then the particle in the front. So in this case, we have a very high energy between the lamellipodia and the nucleus because we want them to be separated. Okay. All right. So for now, we have just kind of copying the old values of the original model into those two new tables, right? But now we have all these other numbers here. They have to decide what values to assign them. And here, so how can you choose them? Okay. So here is a very important step in model building, especially using compartments, is that you do not want to start just adding numbers aimlessly to these compartments. When using cell compartments, you want to have to make a reason out what those values should be before you assign the values. Okay. So first, which should be higher, which should be lower. That's what you have to do. So you're going to do 2 times this, one for the cell crawling example that you're going to do now, and another one for the epithelium. Okay. I saw the question from Clara. I'm going to address that later. Okay. So first, let's start with what should be the contact energies of the nucleus with everything that's external, right? The nucleus is a compartment that have also be inside the cell and never touch anything that's outside. The nucleus should never go out from the outside of the cell. It have always be internal, okay? So because the nucleus has always been internal to the cell, we can say that the energy of the nucleus with every compartment that belongs to a different cell has to be very high. Because when it's very high, then the nucleus is always going to stay inside and never going to move to the other cell. Does it make sense? I hope it does. So this is going to prevent the nucleus from merging with another nucleus or merge with the other cytoplasm or merge with the other cell. We don't want that to happen, so that's why make all those energies very high. Okay. So what about the energy of the other compartments with these other ones, the cytoplasm, lamellipodium, and lamellipodium cytoplasm? Should they be high or low? Okay, answer for Anastasia. Higher than the internal values, okay, you're using rule of thumb, but they don't need to be as high as the nuclear values. Okay, so that's what I guess. Some Rossio said right and 10. Lamellipodium and very low. Okay. So actually, the answer for Anastasia was right on the spot. Okay. They also have to be high because you don't want them to mix, right? But they don't need to be as high as the nucleus with other cells because it's okay for them to touch each other, right? Uh, so you may say that they also have to be high. But it's okay to use a slightly not that high as a nucleus because, okay, if the cells have some kind of adhesion, cadmium adhesion between each cell, each other, right? So they actually have high affinity, have some cadmiums that they bind each other. Maybe you can actually make the uh, cytoplasm, which is going to be the side of the cell to be actually lower than the other. So actually make this low. So this the way the cells actually are attached to each other and they would tend to move together, right? Or maybe they don't share caterings. So in this case, you keep them high. So it has some flexibility about exactly how to do uh, depending on exactly what you're trying to model, right? So here I want my cells to be completely dependent on which one another. So I kept the cytoplasm high, but if I wanted to couple with each other, I make the cytoplasm to be low, and then they touch each other. And of course, remember that cytoplasm is cytoplasm and also the cell membrane around it. Well, so in this case, so this is one way that you can do. Okay. So that's a choice. So you see that we first have to make a guess, a qualitative guess how they should look like. If I want our cells to be kind of independent with one another, I choose like this. If I want them to couple, then I'm going to use this to be low, okay? But not as low as these internal ones, right? Because you don't want them to mix. So it has to be low, but it has to be higher than 10 and 11, okay, for it to work well. Okay, so that's how you do. You first set the two tables, you write on a paper and set what the hierarchy should be. And after you establish this hierarchy, then you go to the code and add the numbers. Okay, so that's this part. So now let's go into practice. Let's now start to change the code. Okay, so now we're gonna do our exercise. We're gonna grab the file that you already have, right? And you're gonna do this 4 different exercises. First, add the internal contact plugin. Specify that all the cells in your simulation belongs to the same cluster. Add cluster division. and go again back to this content, internal and external content energies, and make appropriate choices for them. Okay. So let's start with the first one. So the first step here is to start by downloading and opening the original cell crawling model 6.11a. I think you all have done that, so you can skip to the next part, which is open then intuit it, okay? And take a quick look at it. Actually, we have already done that. See how the model looks like. So you can actually move on to the third part, which is starting to add the ContactInternal plugin, okay? So this is a model that has already been built, and you want to add a new plugin on top of it. So I do not know if you have already done that in the workshop, but when you use the XML file, you can actually go to the menu, CompuCell3D plugins, and specify a plugin and add this plugin to your code. And this plugin is going to be added exactly the same, exactly where your cursor is. So Fatima, yes, it matters where you add the plugin. You first move your cursor on line 53, okay, or maybe a new line here if you need it. And once you are on that line, then you go to the menu, click on CompuCell3D plugins, and then select ContactInternal, okay? Because you want to plug in your plugin outside the other plugins. Don't add any inside another plugin, because then gonna XML is structured, is gonna be confused about what's going on. So please go first, place your cursor at line 53, and then you select your menu and place it right there. And CompuCell3D is gonna write the new ContactInternal plugin for you right there at that point. So please follow the steps here, go to the line 53, leave the cursor there, and then do the clicks on the menus. Has everybody done that? If you have done, please let me know. A thumbs up or just say in the chat. Well, it doesn't necessarily need to be below the plugin, can be above, but you cannot leave your cursor inside another plugin. That's the critical step, okay? Because if inside is going to open new plugin inside a new plugin, and then CompuCell player is going to be very confused, it's going to crash, okay? That's the reason. So it's better to add your ContactInternal after the contacts. Yeah, you can place it on line 37 and paste it. That'd be okay, because line 37 is between two plugins. But you don't want to place your cursor on line 39 to 50, because then you're gonna add your new plugin inside another plugin, and that's gonna give an error. So place your cursor outside of all the plugins. Line 53 is a good choice, and then you do the menu. Does that make sense? Okay, I think everybody is done with that part. Okay, so let's move now to the next part. Okay, you may notice that you place your ContactInternal, okay, and it gives you all your so 3D knows which cells you have, because it can read your cell type plugin and know what cell, so you're automatically going to generate all the combinations, okay? But 3D does not know anything about your model, it cannot read your mind, so you just assign a value of 10 to all those ContactInternal energies, okay? Don't worry about that now, okay? You change them later, okay? What's important is now is that you're on line 53, now we have the ContactInternal plugin, and it's working, so now you can actually start using this idea of cell cluster IDs, okay? Now, before we make changes to the ContactInternal, we have to tell the code that all the cells belong to the same cluster, right? So this is the story that I have already told you before about the cell ID and the cell cluster ID. So you have to grab all those cells in your simulation, which starts from one cell, and tell them that they have the same cluster ID. So to do this, okay, let's go to the code, and we have to go to the steppable file, right? So go to the Python steppable file, and on lines 12 to 20, you have that initial cell volume assignment for the cytoplasm, the lamellipodium nucleus, and that's where we're going to tell each one of those cells that they belong to the same cluster. Okay, is everybody on the steppable files? Give the thumbs up or say yes. Actually, I cannot. It's better if you just say yes in the chat because I cannot see everybody at the same time. Okay, Taylor is, Marcel is there. Okay, Clara. Okay. All right. So if you have any problems during the exercise, please let us know, and someone can go to a breakout room and help you. Okay. So now you go there. So what you're going to do now is going to retrieve the cluster ID of the nucleus, the cytoplasm, which is the first one that you go through, and assign the same cluster ID to the lamellipodium cell and the nucleus cell. And then they're going to be all be three compartments belong to the same cluster of cells. Okay. So the first thing you have to do, we have to first go to make a new line after line 14. So let me go back here. So after line 14, you make a new line, okay? And we're going to use the menu again. Go to CompuCell3D Python, cell attributes, and check cluster ID. So when you click that, you get the cluster ID of that cell. You should get this line here. You may get some spaces as well, maybe get some comments, but essentially what you're gonna get, you're gonna create a variable called cluster underscore ID, which has the value of the cluster ID of the cytoplasm. Did everybody got this? So again, make a new line. So make line 15 below, go to CompuCell3D Python, cell attributes, cluster ID, okay? And then you're gonna get this new line here. Make sure that the indentation is inside the for loop, okay? It may work well, may not work well. If it's not the correct indentation, make sure that's right there, aligned. And by now you should know that Python is very sensitive to the alignment, okay? Now you have the cluster ID of the cell. And now it's time to now use this variable and assign the cluster ID to the other cells. Now, to do this assignment, you need a special command. You cannot simply say cell cluster ID equals something. It has a command that makes this reassignment of cluster IDs. So to do this, now you have to first make a new line after 18, another line under this one here. And you have to go to, again, CompuCell3D Python, cell attributes, and you have this option here called cluster ID reassignment. Okay, so you have to click that, and now you're gonna have two new lines, one inside the lamellipodium and the other inside the nucleus loop, which has self reassigned cluster ID, the cell that you are reassigned the ID, and the new cluster ID of that cell. Okay, so that's how the command looks like. And when you just use this code, you're gonna just get a random number here. I think it's 100 or something else. And you want to replace that number with the variable that you store from before. So you assign the same cluster ID to the lamellipodium as the nucleus, as the cytoplasm has, and the same for the nucleus as the cytoplasm has. So after you do this, what happens now, all those three compartments have the same cluster ID, therefore they belong to the same cell. Does that make sense? If you're done this part, let me know in the chat. Okay. And again, if you need help, let us know. I don't understand how it knows that there are different clusters. Okay, so when you just create the cells and you don't say anything, right? Usually the cluster ID of the cell is the same as the ID of the cell, right? Each cell has its own cluster ID and usually is a unique number, okay? So what you're doing here is we are grabbing the cluster ID of the cytoplasm and assign it to be the same for the lamellipodium and the nucleus. So when you do this, if you have this reassign cluster command, you are changing the cluster ID of the lamellipodium and you are changing the cluster ID of the nucleus to have the same value as the cluster ID of the cytoplasm. Once you do this operation, now all those cells have the same cluster ID number, okay? No, they are already, not means the part in code that separates into different clusters. Right now, you only have one cell and one cluster. You want to actually make all the cells to belong to the same cluster. So actually, we are doing the part where you actually join the cells. Later, you're going to make the cells grow and divide. And then there's a specific cluster-mitosis function CompuCell that when it divides a cluster cell, it makes two new clusters and automatically assigns the same cluster IDs for the daughter cells, for the daughter compartments. Yes. Yeah, now exactly, Clara. Now all the cells in this model belong to the same cluster. Yes. So question from, I don't understand how this would work if you have more than one cell that starts. So because you have more than one cell that starts, right? We create a nucleus, a cytoplasm, and a lamellipodia. Each one are the same cluster, right? So they belong to, so they have different cluster IDs. So what you're doing this loop here is make sure that they all have the same cluster ID. And now they belong to the same cluster. How we're gonna man this if they start with each cell start with as its own cluster, we amend that by doing exactly this code that we're doing here. We are grabbing the cluster ID of one of the compartments and assign that to the other compartments, and now they all merge together. What if you have more than two? What if you start with two cells, right? Made of multiple clusters, right? That's going to be the case for the epithelium. We're going to see how we're going to do that in the next example. Okay. So Kaya, so in the epithelium part, we're going to start with multiple cells, same time with multiple compartments, and then we're going to use a different strategy to assign all the cells in the same column to have the same cluster ID. Okay. Right. Good. So is everybody okay with now, with this part of the code? Yes, yes. Okay. So there's a question from Taylor. Does it matter which compartment has the cluster assignment first? Does it need to be the compartment with the largest volume? No, it doesn't matter. You could have started with the lamellipodia and assign the lamellipodia cluster ID to the cytoplasm nucleus, or you could have started the nucleus and assigned to the others. It doesn't matter. As long as they have the same number at the end, it's okay. So the order in this case does not matter. Okay, does anyone need help? Okay, I see that the mouse from Joanna is already working. Good question from Anastasia. If for some reason, for example, excessive force, the cell splits, right? Would the compartment still maintain the same cluster ID? The answer is yes. They're going to still maintain the same cluster ID. Uh, just because you actually can assign a different compartment that are completely far apart to have the same cluster ID. You can do that. It's going to look weird, okay? But just because the compartments physically separate from one another does not mean that they automatically assign a cluster ID. You have to do that by hand from the Python level, okay? So, okay, good. So I see that some of you are working the model, right? And if you just work the model like as it is, it's gonna not look quite right, and that's exactly the next step of the exercise. Okay, so we did this. Okay, these are all the steps again. If you want to save and play, and if you save and play, you might see that, well, that does not what I expected, right? So Sima shared the same image as me, Luis, uh, Van, Fam, Joanna, yes, that's exactly what I want you to see. Is you shall get something that looks weird, okay? So it's looking weird. It's not looking what we hoped for. What is missing here? You tell me what's missing. No, no, before we make the mitosis, right? This cell here, this simulation here, is kind of working, but it's not working exactly as we expect because we expect it to work like this. And this, well, without the division, right? Uh, and this is definitely not that, okay? So do you know what's missing? What's missing is that we set the quantum internal energies, right? But we did not change the values, right? Remember, we did that step to create a ContactInternal plugin, and it was automatically filled with 10, 10, 10, 10, 10, 10 for all of them, right? And because it didn't change these values yet, that's why your model kind of works, but it's not we have the lamellipodium front, and the nucleus actually is getting outside the cytoplasm, so it does not look right. So what you have to do now, you have to go back to the code, to the XML file, and change the tables, right? The contact energies. So what you have to do, what we have now, we have the thing that you have seen on the top. We have all our energies are still in the contact external plugin, right? And our contact internal plugin that you just created has 10, 10, 10 for all the internal compartments, right? And that's what's leads to that behavior. So what you have to do now, we have to change those values here to be 11, 10, and 40, okay? And that's gonna make your cells look better, okay? So that's what you have to do now. So let's go back to the code, to the XML, and let's copy these values here to these values here. So two things have to do. We first have to copy this cytoplasm from the ContactInternal into the cytoplasm to the ContactInternal, and do the same for the cytonucleus and the lamellipodium and nucleus. Okay, so you can place all this back here. Another thing that you want to do, you want to make sure that the neighbor order is the same for the external and internal one. So if you're using neighbor order 4 for external, you want to do neighbor order 4 for the internal one. The L to L and the cyto and nucleus internal does not matter because you never have two lamellipodiums or two cytoplasms or two nucleus inside the same cell. So all these diagonal terms here in the ContactInternal you can ignore. Okay, you don't have to worry about that. Yeah. You may have a lot of medium in the ContactInternal. If you got that, you can just remove them because the medium can never be internal internal compartments. So if you have medium values here, you can just remove them. Okay. The 3D should not have put the medium there. If it put in your code, you can just remove them and your code's going to look simpler. So yes, remove the medium. So two things you have to do. You have to copy those values here into the ContactInternal, 11, keep this at 10 and make this to 40. And you have to change the neighbor order to 4. Has everybody done that? Yes. Okay. Now that you have done that, did you save and run simulation? Don't worry about the ContactInternal numbers for now. Okay. So if you make all those changes and you run the code, now it looks like this. Now it looks much better. Well, you don't have the division yet, but the cell you should have a cell that looks like this. Okay. Yes, Jenny got it. Okay. Can you explain why you choose 4 and not 1 for the neighboring order? Because I want this neighboring order sets the weight of these contact energies in the overall cost function that governs the evolution of our model. So I want them to be the same as the contact external, so it'll be easier to compare and reason through, okay? So it's gonna just be easier if you use the same. You may have used different neighboring ordering, contact orders for internal versus external. Maybe you want your things to that will you change the relative strength, okay? So it becomes complicated. You may want to do that. You have the ability to do that. But if you do this, it's going to be a little bit harder to directly compare what those numbers mean with what those numbers here mean, okay? So if you make them different, things will get more complicated. So I want to keep things simple. So let's just use 4 here and 4 here, for the same for both. Okay. So please, someone take a look at Kaya's code, see what she did wrong. I don't see anything wrong. make sure you save and run simulation. Maybe it's just that. Well, then stop the simulation of the player and play it again. Sometimes it takes a while to update on the player. Ronex found the issue. Yes. Okay. So did everybody got simulator that looks like this? Yes. Should we move on? Okay, good. So let's implement now cluster division. Okay. So for this, you want to add a new steppable in your code. So let's go to the Python steppable file. Okay. And if you right click at the steppable Python code here, you're going to see that you have an option called add steppable. Okay. If you have not seen this before, this is going to create a steppable for you, right? And update all your files. Okay, so go here and click add steppable, right? And when you have add steppable, okay, you have to give a name to our steppable. Let's say divide cluster cell. And the important part here is you have to select that this is a cluster-mitosis type of steppable. Okay, so that's the crucial part here. So make sure in the pop-up window that you get that you select cluster-mitosis. Okay. The name here can be anything, but here you want to be cluster-mitosis, right? Did everybody got this? So give a name, divide cluster cell, select cluster-mitosis, and then click OK. If you do this, right, you're going to get a new code generated for you, okay? And you see that you have this new code that appear here. And this new code is made of 3 parts, okay? Let me break that for you. The first part here is just to show how you can handle cluster at the Python level. So it's completely unnecessary for the exercise, but it just pays that for you so you can see that you can go over the cells, grab the cluster that the cell belongs to, and then it goes over all the compartments inside your cluster, okay? This red part here is the more important. This is where you decide which cells to divide. It's very similar to the thing as for the division of a single cell. The difference that you are not you're going over the cell and then over all the compartments inside that cell cluster. Okay, and then you have a condition for the division. Okay, and then this last part here is where you do the division, right? Which is just like before, but instead of using the divide cell random rotation, you have the site divide cluster random rotation. Instead of saying this, specify the cell ID or the cell, you specify the cluster ID. Okay. He also generates an import num. Do you need it again? No, there's no need to have 2 import numpy. But having both import functions does not hurt. Okay, so you can just leave it there. But what you want to do here is that we want to, instead of have this division being generated by growth, we want this division to be generated by a random number. So what you want to do, you want to replace this condition for division with the condition if random less than 0.01. So every one step, there is a 1% chance of, 0.1% chance of dividing your cluster. Okay. So what you want to do is that outside the Steppable or right at the beginning of the Steppable file, you want to import the random library from Python. So please add the line from random import star, okay? And then change this condition here. Instead of have if cluster volume bigger than 250, you want to replace that with if random less than 0.001. Please do that and let me know if you have done this. Okay, done. Yes. Now you should have a space after the import. Import, space, and then the star. So this space here is important. Okay. So if everyone has done this, let's move on. And then now your code should look like this, if random less than 0.1, okay? And then what it does is that your divide use the divide cluster orientation. And what it does is that CompuCell3D automatically reproduce the compartment orientation on the two daughter cells. So let's say you take this cluster, choose a random division plane, okay? Cut it in half, and then you see that it has a lamellipodium, cytoplasm, nucleus, and gonna reproduce that arrangement here, lamellipodium, cytoplasm, nucleus, lamellipodium, cytoplasm, nucleus on the two daughter cells, and then they're gonna grow back, and then we have now those two cells after that. Okay. Now with this, we are almost done for the division. The only thing that you have to change is that in the update attributes part, okay, because you're not growing the cell and then dividing, but you just divide the cell and let it grow back, we have to get rid of those two lines. We just copy all the properties of the parent cell to the daughter cell. So you don't need those two lines, you just have clone the cluster cell to the child cell to be equal to the parent cell. So you just have to get rid of two lines here and get on that. So you have to simplify the code, okay? So have you done that? So I'm gonna just show all the comments again, okay? These two lines have to be deleted. Just leave the external energies as before. You're gonna move them as a last part, okay? If you just do this, now you should get a code that the cell's moving, then it divides, okay? But after it divides, it still does not look right. Yes. So Joanna pictures looks exactly like the pictures I have on the screen. Okay. So if you get something that looks weird, I also got something that looks weird. And that's because, yes, the same thing as Ronak. Okay. You're supposed to get bloated. And then the question is why it got bloated, right? Why it looks weird. Anastasia has the answer. Contact energy. We didn't change the external contact energies. Yes. So if it looks weird, that's okay. Okay. I think Anastasia changed the contact energies, maybe. Uh, so yes, okay, she did. No, you don't need. Okay, very important question from Clara. Are we missing the cluster reassignment? No, we are not missing cluster reassignment because when the pump cell makes this division, it automatically knows that it's dividing a cluster. Therefore, you automatically assign a new cluster ID to the daughter cell. So you don't have to worry about that step of reassigning the cluster IDs to all the daughter and parent cells to be different and group them together. You don't need to do that. CompuCell already does that automatically for you. The reason that things went wrong is because we did not choose the appropriate contact energies. So let's go back here and let's choose a new set of contact energies to get a well-behaved simulation. Okay, now this is going to be left as exercise for you. You already know that this one here to be very high. This one here has to be high, but not as much. So you can go to the code and make our own choice, okay? And see if you get a simulation that looks like this, okay? So now your objective, you already have the hierarchy that the contact error should be. Go back to the code and change those values. Anastasia did the change, and you can see here in her picture. And well, the cells are separated, but they're very tied together. If you want them to be more separated, probably Anastasia will have to change them a little bit more. Okay, so if you want to make separate Anastasia, what do you have to change on yours? So if you have to make a choice that gives a nice output, please show in the chat for everybody. A picture and the values that you use. So I'm going to just choose, show the values that I chose. I chose 100 for these ones and 50 for these other two. And when I use these values, I get a simulation that looks like I have here on my screen. If you use those same values, you should get something that's very similar, but it's up to you to make a appropriate choice of these values to get the output that you want. Okay, so here there's no right or wrong, right? Maybe you want to make this very small so that the cells stay together, right? But it's up to you now to do this part. So the Ronak and Rosu make their own choices. So thanks for sharing the pictures. Okay. So sometimes the division, the mitosis division can get some error and can may lose the nucleus. Okay. So that happens very rarely, but it happens sometimes. We're gonna take a look at exactly how we can change the cluster-mitosis plugin to make sure that this does not happen. Okay. So I'd like to move on to the next exercise. Okay. We have done an example of cell compartments using structural compartments. Let's now do example of using cell compartments using functional heterogeneities, which is the case for example, the epithelium, right? So now we're gonna switch gears, right? And you want to make a simulation of an epithelium, okay, where each cell is made of three compartments. They are made of apical compartment, lateral compartment, and basal compartment, okay? And so you can do things like this, for example, to have some kind of apical constriction of your cells, okay? And this apical constriction only happens on the apical side, so you need to have apical versus lateral and basal compartments differentiating your tissue. But that you're gonna learn tomorrow, okay? So right now, we want to do this code, and I want you to all go ahead and download the 6.1.2 zip file, because that's going to be the base code for the next exercise, okay? And then what we're going to do, we're going to first create a model template, adjust cell areas, group cells together, and then adjust the contact energies to have a well-behaved epithelium. So the idea here is just to create a well-behaved epithelium. You're not going to do anything. The epithelium is going to do anything. You're going to sit there, but you need a very stable epithelium first before you can add fun stuff like the epithelium growing, the epithelium have a apical constriction, or anything else, okay? So I have to go a little bit faster here, okay? But now you have a little bit idea of what it takes to make a compartmentalized cell. Okay, so if you want to, let's do the following. Because our time are short, I'm gonna just show the steps. So don't code right now. Leave the code for the lunch and just follow the slides. Uh, just watch the slides and don't code necessarily. Okay. So let's follow this one. So for example, if you want to do now this new model, let's go CompuCell3D project, create a new project, okay, give a name to the project, okay, tissue folding, Python XML. Here, what you're gonna do, you're gonna change the dimensions of the model to be 350 on the X dimension and only 100 pixels on the Y dimensions. And you want to make it periodic along the X. So you have no flux here, you're gonna change the X to be periodic, okay? And you're gonna leave this as rectangular step and hit continue, okay? And next, what you want to do, you want to add the 3 compartments and plus an external compartment, an external cell that's gonna be the ECM. So you want to add apical, lateral, basal, and ECM. So you're gonna have these 5 cell types, including the medium, okay? So you have to do this, hit continue, okay? You're gonna ignore the chemical fields. And here on this Polarity simulation wizard, because they're creating a model from scratch, okay, we have to make sure that we select ContactInternal. That tells your wizard to actually create a ContactInternal plugin. So you have those 2 tables, the external contact energy and the internal contact energy, right? So make sure you select that, hit continue, is done. And you can either do this or you can only download the 6.1.2 zip and already have all the code, this part done for you, okay? So let's move on. So what you want to do now, you're gonna create this following initial condition, where you have create a line of apical cells here, a line of lateral cells here, a line of basal cells here, and a line of very big ECM cells at the bottom, okay? So usually I leave this as an exercise for you to do, but you can just look at this and think, okay, how are you gonna make this initial condition? To do this initial condition, you basically need to go to the uniform initializer and use the region code 4 times, 1 for each part. You're gonna do this, for example, box minimum 0 to 150, so spanning all the X dimensions, and the Y dimensions you made it from, let's say, 49 to 56, okay? And then this code, you create a series of apical cells, all of with 7 by 7 pixels. And then you can use the same trick, the code to create the lateral line of cells, and the same code to create the basal line of cells, and the same code to create the ECM line of cells right at the bottom. So if you do this, your code is going to look like this, okay? So you have an initializer, you have this initial line that creates the apical line, this other one, again, from 0 to 250 to span out the X dimensions, but at a different Y coordinates to create the lateral cells, another one here to create the basals, and this other one here with a weight of 35 to create the ECM on the bottom, okay? So you don't need to code that. You can just download the file 6.1.2 and you have this implemented, okay? So if you do this, or if you just download this code, unzip and play, you may see that you get the initial condition very briefly, but everything looks fuzzy because when you just use an initializer, each cell is its own cluster ID, right? What you want to do now is specify that all the apical, basal, and lateral cells that have the same Y coordinate at the beginning, they should belong to the same cluster. So you have to reassign the cluster ID. And you have to do this for multiple cells at a time. So that goes back to one of our questions at the beginning, how you can make this cluster ID assignment, right, when you have multiple cells or multiple compartments to group into. Okay. In this case, we can use the symmetry of the tissue to make this assignment. So you have to group the cells together, and this has to be done on the Python level, right? So how are you going to go about that? You basically use the start function of your Steppable. You're going to check all the compartments that they have the same Y center of mass, or sorry, X center of mass, and if they have the same X center of mass, you group them together. Okay. So, so one way to do this, you can do a looper of one cell type. Inside, add a second loop over all the other cell types. And if they match the center of mass in the X position, you can reassign the cell ID. Okay, so that is an algorithm that you can use. Okay, so this is the algorithm implemented. Okay, we start with the apical side. We do a loop of all the cells of type apical. Inside this, you do, you don't want to add the ECM there. So the question, how do you add the ECM this? Your ECM is going to be a class of cell type that you don't want to group. You want the ECM to be its own thing and not be a part of a group of cells, okay? So you leave the ECM out, okay? So let's go back to the code here. So here I'm just implementing the algorithm. This is the algorithm that I'm going to do, or this is the pseudocode, and this is the pseudocode implemented on CompuCell. A loop over all the cells of type apical. Then you do a second loop of all the cells of type lateral. Pay attention that here you have to assign a different dummy variable. Here was cell. This is going to be cell 2, right? And then if the cell 2 has X position equal to the cell 1, I reassign the cell 2 to have the same cluster ID as the initial cell. So this is the cluster ID of the cell, the first loop of the apical side, and assign that value to be for the cell ID. And once you find that cell the same position, you can stop the loop, you can break and get out, and you proceed to the other loop over all the basal cells. And again, you check again if the X coordinates the same, and if they're the same, you assign the cluster ID of the apical cell to the other cell. And once you do that, you can break. You don't need to check it again. Okay. Once you do that, now you can see that all the cells belong to the same cluster. Okay. And you can visualize that in the player. If you click on configuration, you may see that you have an option for the color of the cell border and an option for the color of the cluster border. Okay. So you can assign different colors for them, so you can actually better see when the cells belong to the same cluster or not. What I like to do is I like on the player to draw the cluster borders, but not draw the cell borders. In this case, this board between the compartments that belong to the same cluster is not drawn. You only draw the borders of the cells that belong to the different clusters. I like this visualization better. Okay. But you can make, for example, the cluster border to be white and the cell border to be black. In this case, you're going to be white outside here and black in between. Okay. So again, you don't need to code this. I'm just showing this. You can just load this file 6.1.2c zip, and you already have that implemented for you. Okay. Does anyone have a question about this? Does it make sense? If so, please tell me on the chat so I can know that how things are going. Okay, so all good, makes sense. Where did the code occurrences go? Okay, let me just show you this. Okay, no, sorry, I have to show in the player. Okay. So here, if you click on visualization, what I like to do, I have the cluster borders marked and the cell borders unmarked. Okay. The reason I do that, okay, is that when I make this choice here, I don't draw the cell borders, so you don't see a border between the cells once they're grouped, right? But I see the border between the clusters, okay? So here you're selecting what's going to visualize and what's not going to visualize. And then if you go here to this configuration and go to the cell colors, you see that you can choose the color of the borders. So I chose the cluster borders to be black, for example. So that's why you see a black line around here. If I chose this color to be, I can click here. Where is it? Okay, I don't know where it go, but usually when you click this, you can choose another color for these boundaries, and you can make your boundaries to be black, white. That's going to be you, two colors. People choose can make it to be pink, wherever you choose. So that's how you choose the colors for your borders. And here is where you choose which one to show or not. Okay. Good. Is it possible to fix the basal cells to be mobile? Yes, it's possible. But you can, for example, assign a very big lambda volume to them, and then you'll never make any pixel change, so they're going to be fixed, right? Finally, someone using Mac. Yes, I'm a Mac person. Okay, go back here. So you have this code here, and now is the part that's very important. Now is the part that I really want you to do, okay? Which is, so far you have just this did, right? The cells are compatibilized. You have apical, basal, and lateral belong to the same cell. But if you just run the code, nothing really happens. All the compartments have the same value. Contact energy is all 10 in your code, right? Now it comes the part where you should be trained as a modeler on the correct use of your contact energies, right? So you have to choose what values of the contact energy should be and what values for the internal contact energy should be, in order for this to be stable, okay? So now, remember, what you do first? Do you just start adding numbers, or you should do something else? You don't start adding numbers at random. You first work out the hierarchy of the contact energies. What should be high and what should be low and what should be something in between the high and the low. So you first choose the hierarchy of values. So you first design the tables in a qualitative manner and sort out the hierarchies, and then you start adding numbers, okay? So let's do this. So where do you like to start, okay? So first you do this, and the rule of thumb, remember, is one set of values should be higher than the other set of values in principle. Which should be higher and which should be lower in principle? Should this be higher or should it be lower than the contact internal? Very good. In principle, the contact external should be higher than the contact internal, right? So that's the rule of thumb. And why should it be like this? Why I want this to be lower than that one in general? Osmond gave an answer. Is that the correct answer? Yeah. Yes. Yeah, so both answers are correct to prevent mixing, so they don't mix with the medium as well. Uh, so usually what belongs to the same cell should be more compact, right? Because it belongs to the same cell. So you want those compartments to be more close, tied to each other, and less tied to the things that belong to other cell, right? So usually these values here are lower than these values here in principle, right? So we already have these hierarchies set. Let's sort out. But now let's work out how this should look like. Okay, so this is going to be a very important exercise. Okay. Uh, so let me go back here. So let's start with we want these compartments here to be cohesive, right? So we want to set this energy low. Do you agree with that? So yes, does make sense? So okay, so this apical lateral, apical basal, and lateral basal should be low, okay? There's one of these pairs that can be high, can be a little bit higher. Which of these pairs can be a little bit higher if you want? Apical basal, because they are different parts, so actually you may choose apical basal to be maybe a little bit higher, right? So a low plus or medium if you want to, okay? So that's okay. But in principle, they have to be low, right? So let's start with that. Let's start simpler, okay? And what else can you say about this contact internal plugin energy? Very good question for Anastasia, okay? They should be low, but she asking, they shouldn't be too low, though, because we still want the spatial organization to remain. So yes, that's completely correct. If you make them too low, if you make them zero, what will happen to these internal compartments? So if I make this, instead of low, I make them zero, they'll be complex mix, right? What if I make them negative? If I make the negative, they become even more mixed, right? Now, they'll not necessarily disappear, but they'll become really mixed with one another, okay? And that's actually something that you may want to desire, okay? That's actually a technique that you can use to make your compartments dynamic. So you make all your internal compartments energies to be negative or zero, okay? So they're all mixed, okay? And then what happens? So how can you get anything if you have all your compartments mixed? Well, you still have the external contact energies. So when the cells start interacting with something external, these external interactions can then mix the compartments, okay? So you can have a cell that can easily adjust its polarity under the influence of the external environment or the other cells. So that's something that we use, for example, to model planar cell polarity. Okay, we have the cells internally, they are mixed, but the external contact energies are positive, and this external contact demix the cell. Okay, so uh, so that's something that you can use. Actually, is a technique that you can use to make things more dynamic. Okay, so for example, that's what I use. For example, also in go back very quick here. That's what I use here. And also that's what I use to model this one here. You see that they're kind of mixed, but once they have contact with other things, they become well separated. Okay, so that's something that you can do. But let's go back to this code here. Let's first learn how to use this the correct way. Okay, so what about this ECM column here? Do you have to worry about this or not? No, because ECM will never be part of the cell. It's something external to the cell, so you can ignore that. What about this part here, the contact internal with the medium? Do you have to worry about that or not? I also know because medium is never a part of the cell. Okay, so you don't have to worry about that. What about this diagonal term here? Do you have to worry about this or not? Why not? Because you only have each one of them. So therefore, you can ignore this and this. You can also ignore that other one because we never have repeated compartments inside the same cell. You may have repeated compartments, that's legal, you can do. So there's nothing that prevents you to do this. Sometimes you want to model a multinucleated cell, for example, right? Or if you're modeling the mitochondria inside the cell, you have more than one. So maybe you won't have, so in this case, you have to specify these diagonal terms, okay? But most of the situations you don't have to, so you can ignore. So we are done with the contact internal. So we can move on to the contact external one. So let's start with the diagonal terms, how they should look like. Okay, they should be low. Should they be as, why they should be low? So when you specify this contact energy, is the contact energy is this apical side with this apical side, okay? So you said that should be low. Why should it be low? It should be low because you want them to actually align with each other, right? And the same thing for the lateral. You want them to be aligned with one another. You don't want your lateral to go up and you want your lateral to go down. You don't want your base to go up or to go down. So you basically choose this apical, lateral, and basal to basal to be low. So they actually, the basal align with each other and the apical align with each other and the lateral align with each other. Does it make sense? So you usually want to be low. So should it be as low as this, lower, or a little bit higher? a little bit higher because they're not internal compartments, right? So you're going to want to run this here to be low plus, okay? So let's look at low, but higher than the other one. So let's do right here, low plus. Does it make sense? Okay. Now, in the same idea, if you want this to be low to make them kind of align with each other, what about this cross terms? What about apical with lateral and apical with basal? So, or lateral with basal? Or what about this cross term, this three cross term here? How they should be? Uh, Clara asked a question, would that depend on your neighboring order that you use for the content error? No, it does not depend. This hierarchy is completely depend on the neighbor order that you use. So I would say that's going to be a little high, maybe medium, okay? So it's high, but not that high, because you kind of want to penalize when this goes up here. So for example, this content here, the green content, the medium, you don't want that to happen that often, right? You may want to happen a little bit, it's okay, but you don't want to this green red completely go over here. That's going to be bad. They're going to disalign your epithelium. So in principle, I agree with most of you, it has to be high, okay? Okay, so in principle, it has to be high, or maybe medium. Now we have all these other terms here. What about the contact with the ECM? Okay, what can you say about this column here? Should be high, low, or should have a mixture of high and low? Low with A and high with the others. Do you all agree with that? So let's start with the two extremes. Let's start with the ECM and medium energies. Should they be low, high? Do you want them to be in contact with one another or be completely separated from one another? Yeah. So this they want you never want this to go there. So you want to this want to be very high, right? You may add high with bold letters. What about the ECM and the apical side? Should it be high or low? High, because you want your apical side to be not touch the ECM, right? So you want to be high. And what about, oh, sorry, I kind of skip a part here. Let me go back. What about the apical side of the medium? Should that be high or low? So you don't want the apical side to touch the ECM, so you want that to be high. What about the apical side in the medium? You want your apical side to touch the medium, right? The apical side is the part of the cell that actually touches the medium. So actually, you want that to have a low energy because you want that to be touched here. So uh, you actually do high here, but low here. Does it make sense? And then you can apply the same logic for the B, for the basal side. What should be the energy of the basal side of the ECM and what should be with the medium? It should be B ECM low, okay, and B medium high according to Hipla. And I think she's correct. I agree with that. That's actually what my choice. So because that makes the basal side to like to be in touch with the ECM, but not like to be in touch with the medium. Does it make sense? I think it does, right? Okay, now what about the lateral part? That's the one that's lacking. Should it be high, low? What should it be? You can make it high, yes, because you don't want it to go to the medium and you don't want to go to the ECM. So you can also make that high. Okay. All right, good. And then what's left? The ECM and medium have to be very high. And what about ECM with ECM? Well, ECM, we can actually mix those two, so you actually can make that 0 if you wish, okay? Because you actually can model that as a single cell, but it's going to be harder to do that in the niche conditions. So actually you can make, this is an example where you can actually use 0, and that's going to be completely fine. So with this, we finalize our table. So our table is completely qualitative, right? But it has the correct hierarchies, right? And now that you have chosen these hierarchies, now it's time for you to go to the code and implement that and add the numbers on it, okay? So you can do that now as an exercise, all right? Lunchtime, you can do this, okay? I see that Kaya has already made her own choices and she has a stable epithelium. And the idea is that you choose some kind of values that obey the hierarchy. You may have to adjust a little bit, but why is it important to do this qualitative first? It's important to do this qualitative first because, as you can see, these are a lot of parameters. It's not something that you can get right by just trial and error, right? You cannot kind of move this up here, here a bit, and maybe you're going to get there. It's going to take you forever to get it correct if you don't reason the values first. So it's very important to reason those values first. And this reason is not hard. I mean, look at what you want to be, look at the code that the model that you want to have, what your cells want to look like, what you want them to behave, what rules have to obey to be stable or for the cell to move in a certain way, right? And just looking at the biology, you can kind of already guess what those values should be, right? And you can come with this hierarchy of values for your additional energies. And then as you work this out, it's good because first, when you do this exercise, you learn more about the biology, right? You know what should do. And then it's going to be much easier for you. You add the numbers. Maybe you made the numbers out very close to each other, so there's not much difference with them, so everything's going to look the same. Then you can make it a little bit higher. So when you have high, you go to 100. When you have low, you go to 5. When you have low plus, you maybe go to 10. When you have high, that's not that high, maybe go to 50. So then you start to play with the numbers and you get something that going to look like. And then once you get something that looks good, you maybe then start to do the fine tuning. Okay, maybe I want to make this a little bit higher. Maybe I want to make my lateral to be a little bit lower because that's where my cadrins are. Okay, or maybe where my desmosomes are. So maybe then lower. Okay. Uh, but basically that's it. This is just an example of the numbers that I chose and give a stable epithelium. That's how it looks like. And you want to look at this code, you can just download 6.1.2 zip. But that's basically what I have for now. I'm going to stop here and I'm going to just tell you that you should get a stable epithelium because what we're going to do tomorrow, you're going to add forces between the apical cells to get further formation. So that's going to be for tomorrow. But I'm going to just stop here and ask you to fill up the questionnaire, okay? And if you have any questions, I'll be happy to answer them while you go for lunch. There was a question in the chat. What's the difference between the ContactInternal plugin and the Contact Compartments plugin? Pedro has already answered. The Contact Compartments is deprecated. Okay, it's not used anymore. That was the old one. Don't worry about it. Just use the ContactInternal. That's the one we use to run the code. So Anastasia asked, the epithelium collapsed toward the ECM if I try low values. Collapse like the ECM is disappearing because we didn't set a volume, or it just goes into the ECM. So if the ECM is disappearing downwards, Yeah, you may have not forgot to set the ECM volume. So if you code everything, you may have missed a step. Yeah, it may be that. Try adding a volume to the ECM. Joanna, I think your simulation looks fine. Set the, you see that you have this kind of domes. That's because the ECM is not mixing with one another. If you go back to your ContactInternal, set the ECM to be ECM energy to be 0, you're gonna see something that looks more like in my picture here.